Hello everyone, welcome back. Today we're going to start Lab 2, which is the polymerase chain reaction, or the PCR. So you guys can follow along with me on page 14 of your lab manuals. So to begin, we're going to start by adding 19 microliters of DI water into an empty PCR tube. Next, we're going to add our primer. The first one will be 2.5 microliters of our reverse primer. And then 2.5 microliters of our forward primer. Then we're going to add 1 microliter of our DNA template, which for us is the beta-gal gene. And finally, we're going to add 25 microliters of our Q5 master mix. So this master mix contains all of the buffers and the salts needed for this reaction to occur. So I'm going to add it into the tube and then kind of go up and down with the pipetter to mix properly. And there we go. We have 50 microliters of our PCR reaction. So next, what we need to do is add it into a thermocycler so that the reaction can occur. So that is what this green machine is over here. So if you guys look right over here, you can see all of the conditions that will be, I guess, all the different temperatures and cycles that the PCR reaction will go through. Starting with the left, we have our initial denaturation at 98 degrees for 30 seconds. Then we hit this middle block, which is the actual cycles part. So we have our denaturation step at 98 degrees for 10 seconds. The annealing of the primers at 67 degrees for 20 seconds. And then the extension of the gene at 72 degrees for 1 minute. And you can see that this is going to go for about 30 cycles. Once these 30 cycles are complete, then we're going to go for a final extension at 72 degrees for 2 minutes. And then once that is done, our PCR reaction will be complete. So I'm just going to add our PCR reaction into the thermocycler. And just run the protocol. So while this is running, we can start with our next step, which is to make the agarose gel so that we can kind of analyze our PCR reaction to determine if the reaction was a success. So in a small little bottle, like this one, I'm going to add 0.26 grams of agarose, as well as 35 milliliters of that TAE buffer we made last week. All right. So now just kind of put the cap on and give it a little swirl to mix. So I'm going to tighten the cap, but not all the way. Just make sure that it's a little loose, because the next step is we're going to put it in a microwave. So we need to heat up this reaction, I guess kind of thoroughly, to dissolve all the agarose, and then let it cool so that the gel can polymerize. So I want to make sure that the cap is loose so that while it is cooking in the microwave, it doesn't explode. So I'm going to let it go for about 15 seconds. And this should help get the agarose powder to kind of dissolve properly. And once it's done, I'm going to continuously look at it, see if there's any particles in the solution. If there are, then I just throw it back in the microwave. So let us see. So it's done, and you can still see that the powder hasn't completely dissolved. So again, I'm going to throw it back in. Go for another 15 seconds. And I'll see what happens then. Alright, so it looks like the bottle is starting to boil, or at least the solution is starting to boil. You can see that the powder is starting to dissolve a bit more. So I'm going to put it in for another 10 seconds, and I'm going to keep reducing the time until eventually we hit about 8 seconds, and then I'll keep going at 8 seconds until the powder is completely dissolved. So you can kind of see the bubbles at the top now that the solution is definitely boiling. You see that the powder is gone, but if you look very closely, it's really hard to see on camera, there are still these little particles inside of the solution. So these are the particles I'm going to try to dissolve completely. So this process can take a little while, so we'll get back to this when the solution is completely dissolved. Alright, so I've already finished microwaving the solution and I was letting it cool down. So while it was cooling down, I covered this gel tray with tape. So the reason we put this tape on here is so that when we pour our agarose gel into this gel tray, it'll be able to solidify properly and not leak out. So now that this bottle has pretty much cooled down, that I can kind of hold it in my hand, I'm going to add 3.5 microliters of a dye called Cyber Dye into my solution, into my gel, so that when we look at this gel under a blue light, we'll be able to see our proteins. So just give it a little mix. Put in our gel comb into our gel tray. And now pour in the agarose gel. So if I made the tray properly, nothing should leak out. And now all we have to do is just let it sit until it hardens. So I will get back to you guys when that happens. Alright, so skipping ahead a bit to the future. The PCR reaction has finally finished, and this is what I ended up getting. So now it is time for us to set up the gel to see if the PCR reaction actually worked. So I stored the agarose gel in the refrigerator, where you can see that it completely solidified, nothing comes out. So now we are ready to get everything set up. So the first thing I'm going to do is remove this tape. That way I can put this in the gasket. All right, so now that all the tape's removed, I can place it into the gasket. Remember that wherever the ladder is, it should be closest to the black side or the negative side. And the reason being is that our DNA is negatively charged. So when we run the current, the negatively charged DNA will want to move towards the positive side of the gasket. So it's going to move down the gel. So now that I have my gel inside of the gasket or the chamber, I'm going to fill it up with that TAE buffer we made last week. So I'm just going to fill it up till it hits this max line on both ends. So because there's like two little chambers here. All right. So now that I've added the TAE buffer, I'm going to put one hand down on the, I guess the bed of the gel tray and remove the comb by kind of wiggling it out until it eventually pops off. And what you should get is something like this, where you can kind of see that there's little indentions of what to add into the gel. So in the very first well, I'm going to add about 10 microliters of a DNA ladder, which is this blue solution right here. So I'm going to be very careful to make sure that I add it in and don't stab all the way through. And just let the solution kind of leak out into the well. And then next, I'm going to add 10 microliters into the well right next to it so that we can analyze it once the gel is done running. Same thing, I'm going to be very gentle and make sure that my tip kind of hovers above the top of the well and slowly drop my solution into the well. And so while you can't see our PCR product because I didn't add a dye to it, you can definitely see the ladder that we're going to be using in the first well. So now that that is set up, I can place the lid onto our chamber. And I'm going to let this run at 120 volts for 30 minutes. So if you look at it closely, I don't know if you can see it through a camera, you can see that there are bubbles running through the sides, like right here, to signify that there is a current going through the buffer. So I'm just going to let this run for 30 minutes, and then I will take a picture of what the gel looks like and post it on Blackboard for you guys to use on your report. So remember that for this lab, we will finally start writing our reports, as well as also take a lab quiz. So I hope you guys have a good time with this experiment.